The effect of deuterium depletion on the 2nd of June gene expression. Experiments carried out in the past years have revealed that cyclooxygenase enzymes and the prostaglandins synthesized by them have an important role in the regulation of tumor growth and metastasis. 18, 19, 20. Prostaglandins are formed from arachidonic acid in reactions catalyzed by phospholipase A through cyclooxygenases. Latest research has revealed that the enzyme cyclooxygenase has two isoforms: cyclooxygenase cell COX-1 or PGH1 and cyclooxygenase 2 COX-2 or PGH2. COX-1 is bound to the cell membrane and to the membrane of the endoplasmic reticulum. Chemically, it is a molecule of hemo or glycoprotein nature. It is produced continuously in almost every cell. Its primary function is to mediate the synthesis of prostaglandins needed for the normal functioning of the cell's housekeeping function. Its quantity is nearly constant. COX-2 is not found in inactive cells or in very small quantities only. Induced by growth factors, endotoxin, and cytokines, its expression grows dramatically, a 10 to 80-fold increase in activity. During inflammation and cell growth, this happens at a very early stage. Its amino acid sequence is to 60%, and molecular mass to 100% identical that of COX-170 DAW. On the basis of the aforesaid, it is obvious that the two isoenzymes have different intracellular functions. Division and other inductive processes are generally connected to the activity and expression of COX-2. Several experiments have proved that tumors synthesize. Figure 11.1 The division of two cell lines. HT29 human colon carcinoma and normal primary myometrial cells, HMC, in culture media with different deconcentrations. More prostaglandin than healthy tissues. 18. It can, thus, be supposed that COX play an essential role in tumor formation, in the regulation of tumor growth, and in the development of metastases. This is confirmed by experiments where the inhibition of prostaglandin synthesis by non-steroid anti-inflammatory drugs and NSAIDs caused tumor regression in the case of colon, breast, esophagus, lung, and oral cavity cancers. 20. The higher Pg concentration in tumor cells is the effect of the increased synthesis, for which the COX-2 isoform is responsible in the first place. Because of the possible cause and effect connection between the overactivity of COX-2 and the higher Pg concentration on the one hand, and the induction and metastasis of tumor on the other, it seems that targeted inhibition of COX-2 could be an effective approach in preventing cancer. Experiments were carried out with normal primary human myometrial cells and with a tumorous cell line of human colon origin adenocarcinoma HT29. Human myometrial cells were obtained after the digestion by collagenase of uteruses removed surgically in hysterectomy. Both cells were cultured in RPMI media containing 10% fetal calf serum FCS in a carbon dioxide thermostat 5% C2, 95% air. When cells had almost filled the space in the culture dish, 70% confluence value, we withdrew the serum for 24 hours. Following serum deprivation, the cells were given RPMI media prepared with deuterium depleted water, and after 24 hours the NTT microculture tetrazolium assay was performed to determine cell proliferation. Figure 11.1 shows the division graphs of the two cell lines taken as a function of deconcentration. The figure shows well that the D content of 20 ppm inhibited in 45% the proliferation of HT29 cells. Inhibition was connected to deuterium concentration. The degree of inhibition decreased continuously, and at a deconcentration of 60 ppm it approached the value measured at normal 150 ppm deconcentration. In the domain above normal deconcentration, 500, 1000 ppm, cell division was again inhibited. The figure also displays that the effect of deconcentration was minimal on non-tumorous myometrial cells. The above experiment, 2, reinforce our previous statement that deuterium depletion has but a minimal effect on healthy cells. Figure 11. To demonstrate the connection between COX-2 gene expression and deuterium concentration. With Western blot analysis, we measured the COX-2 content of cells treated as detailed above. Assay has shown that DD water 20 to 60 parts per million strongly inhibits the COX-2 gene expression of HE29 human colon carcinoma cell lines. Similarly to the results of cell proliferation experiments, here too, the effect proved to depend on the concentration. Figure 11. 3 demonstrates the effect of deuterium-depleted water on COX-2 gene expression in cells, both basic unstimulated and stimulated with cytokinase-1, in human myometrial cell lines. As an effect of DD water, expression was inhibited in the case of unstimulated cells as well. But the inhibitory effect of D-depletion was especially expressed after stimulating the COX-2 gene expression with interleukin. The results confirm our earlier observations that DD water inhibits COX-2 gene expression. The above results suggest that the decrease in deconcentration influences the COX-2 gene expression and the activity of PG formation in cells. The PG synthesis of cells is significantly inhibited by deuterium depletion. In normal 150 ppm water, the P level was 242.9 mol, whereas in 20 ppm DD water it was 76 cg/mol. These findings confirm earlier results obtained during the examination of apoptosis. We suppose that increased apoptosis induced by deuterium depletion is realized via the inhibition of COX-2 gene expression and PG formation. The effect of deuterium depletion on the hydrogen ion regulation of cells, a given molecule of element, can play a regulatory role in the cell if its concentration change is in a way perceptible to the cell, triggering or halting specific intracellular processes. It may be supposed that behind the regulatory role of deuterium there lies the fact that the intracellular DH ratio can change. Taking into consideration our knowledge of discriminative processes connected to deuterium, we suppose that the pH regulation of cells and processes happening through the cell membrane might be connected with the change in the intracellular DH ratio. Thus, we examined whether the decrease in deconcentration influenced these processes. Experiments were carried out with a 4-murine hemopedic cell lines. Intracellular pH was artificially shifted into an acidic direction, and we then investigated how pH is stabilized again in the media of normal and of depleted deuterium content. These experiments testified that in the media of depleted deuterium content, intracellular pH returned to the normal value much slower than cells of normal D content. It also became clear that in this process, the amiloride-sensitive Na+/H+ antiport system plays a key role. This observation points to the fact that enzymes and processes, well known in transducing signals and pH regulation, may be sensitive to the change in deconcentration, which raises the possibility that their activity and functioning can be influenced by the change in deuterium concentration. The biological effects of D-depletion on plant cells were investigated in the leaves of the Canadian seaweed Delodia canadensis. It was possible to follow several biological processes after having placed the leaves into a DD water medium, measuring respiration, photosynthesis, membrane potential, and changes in pH, both intra and extracellular, unanimously pointed to the fact that in the first half hour of deuterium depletion, the plant showed such biochemical changes as if it were put in the dark. its respiration became quicker. Photosynthesis stopped and intracellular pH became alkaline, whereas extracellular pH changed into an acidic direction. 14. These effects were at a maximum some 30 minutes after treatment. Reactions gradually stopped later. All the above makes us conclude that plant to perceive and experience as a stress if the deconcentration of the medium is decreased. In time, cells adapt to the altered circumstances and the stress reaction gradually ceases. All this raises the intriguing possibility that in cells, both vegetable and animal, there exist such mechanisms which are able to perceive changes in deconcentration. This induces a stress reaction of some kind which, in turn, contributes to the adaptation of cells. Taking into consideration the high degree of sensitivity experienced in tumors in a medium of decreased deuterium content, we may suppose that in tumor cells this adaptation happens slowly or not at all, which results in the necrosis of cells. The effect of DD water on the germination of seeds. It is a well-known fact that following water intake, a very quick growth begins in germinating seeds. The effect of DD water on the germination of seeds was interesting from two aspects. The first is that this is a very simple, if not the simplest, system to examine the effect of DD water in. And the second is that it can provide further proof that effects observed in animal cells can be observed in plants as well. We performed germination experiments with several plant species and about 12 to 14,000 seeds in standardized circumstances, in a medium of 20 to 300 ppmd content. It can generally be stated that in a medium of depleted deuterium content, in all independent experiments, the sprout and root lengths of the seeds were less compared to the controls, as shown in Fig. 2. 3. We experience a significant difference in sensitivity between the plant species and also between breeds. While in the case of oak, the inhibitory effect of DD water was a mere 5% compared to the control. With squash, this was 60%. 12. 14. 21. Regarding the scatter of the experiments, a deviation greater than 10% from the control group can be considered as significant. Examining the effect of decontent on the germination of rice, we may conclude from the results that the optimal deconcentration water for seeds falls into the natural 150 ppm range. It is important to stress that inhibition was at its strongest 5 to 6 days after the beginning of germination. Having measured sprout length after 10 to 12 days, the effect experienced earlier could not be observed. This confirms the results. Table 11. 3. The change in the length of plant sprouts in normal and in depleted water. Of experiments carried out with heavy water, where it was stated that living organisms are able to adapt to the growing decontent of the medium within a relatively broad range. According to our results, cells are able to adapt also to Table 11 for changes in the sprout length of rice as a function of the decontent of water, the depleted medium relatively quickly. This observation confirms our results with in vitro tissue culture, where a more significant effect was experienced at the beginning of the treatment.